Our immune system has a unique ability to recognize diseased or malignant or transformed cells and distinguish them from healthy tissues. There is a very particular and unique subset of immune cells called T cells that can recognize through T cell receptors different molecules that are different in healthy and diseased, infected or malignant tumor tissues. The classical T cells or conventional T cells that recognize peptides are able to distinguish the mutated proteins or peptides derived from tumors, and through this recognition distinguish them and eventually kill. But the problem is that a lot of, or most of the pediatric tumors and many adult ones, they come with low mutational burden and therefore low immunogenicity. So the T cells have a very limited number of these mutated peptides that they can actually use to recognize tumors from healthy tissues and then to kill and eliminate those ones. In contrast, most of the tumors have a drastic metabolic perturbations, and that allows a specific subset of these cytotoxic T cells called unconventional T cells that recognize small metabolites to more efficiently recognize and distinguish tumors or infected cells from the healthy ones. We want to study the activity of these cells and learn how we can manipulate their activity in order to enhance their ability to kill the cancer cells. One of the unique features of these cells and these metabolite recognitions is that those metabolites are present in many different types of tumors, so they are very common. And they are not present in healthy tissues and organs. Therefore, this provides the very fine specificity and activity of these cells that can sense the tumors but do not recognize and harm the healthy cells and tissues. We have two focal points. The first one is on the T cells that are specific for the tumor metabolites, and the second one is on the tumor metabolism. So the first focal point comes with the first goal is to identify the tumor-specific T cells and T cell receptor that we can then clone and use for the immunotherapeutic approaches. If we find the receptors, and we know that these T cells and receptors exist that can recognize a variety of different types of tumors without touching healthy cells, we can find a golden bullet to actually target many different types of tumors independently of their origin, the tissue they originate from. The second focal point of the project is the metabolism of tumors. So there are already a number of drugs that have been used for the chemotherapies in children and adults. We know that most of these drugs induce a drastic metabolic perturbations in tumor cells, but also healthy tissues. So in these regards, we want to screen and find the drugs that in a very specific way change the metabolism of tumor cells, making them more immunogenic. So the idea would be to have both in one shot, where you provide a drug for the chemotherapy, but at the same time, along with the killing of the tumor cells using the specific drugs, so drug combination, you would also induce the activity of the specific T cells that would help to eliminate these tumor cells or avoid metastasis and kill those tumors more efficiently.